All right, welcome back, everybody. So we've got our final lecture in our fall semester series on our urinary system. Okay, so so chapter 18, talking about the urinary system and fluid balance. The nice thing about this system is it's fairly simple in organization because the anatomy is really focusing on four main structures. But the physiology is pretty neat because it's going to be fairly expansive and fairly encompassing. And we'll see that we've got a lot of other systems of the body that are going to be at play, that hormones and things that the kidneys, as well as some of the functional components of the kidneys produce, can act on various other elements of the body. So let's go ahead and get started here. Okay. So the kidneys have six major functions, right? So we typically refer to the urinary system also as the excretory system, because mainly the excretory system is responsible for getting rid of waste, right? That's kind of how we think of it. So with our excretory system, it's not going to be just for excretion, right? So we also have a bunch of other things that are going to be involved here. So one of the primary functions of the kidneys is actually regulation of fluid volume in our body, and by association, things like blood pressure. Because as we understand now, our blood has a significant portion of it that is wrapped up in water content, along with things like plasma and other kinds of proteins. And it's not just erythrocytes. So the kidneys play an important role in regulating blood volume and blood pressure. Excuse me. Not only that, but one of the mechanisms by which we can control that is by figuring out how many salts or how many kinds of electrolytes or other kinds of nitrogenous waste that we have floating around in our blood, and then we can excrete that and get that out of our body if we need to as well. So we can control things like the pH of our body with our excretory system, right? And then we have things like regulation of red blood cell production. One of the main hormones that the kidneys produce is something called erythropoietin. So erythropoietin, if you guys remember from way back in chapter 11, that's the main hormone that triggers the production of red blood cells and stem cells from red bone marrow, right? And we also utilize the kidneys as an important precursor for vitamin D synthesis with respect to concentrations of things like calcium ions in the blood, right? Let's see. So yeah, Catherine, just really quick. This lecture is being recorded right now. This will get posted on YouTube immediately after class tonight. The other two lectures, so chapters 19 and 20, as I'd already mentioned, those are going to be, I thought I had posted them, but Devin had said that they didn't get uploaded for some reason. But I have them recorded already from a previous semester, so those will get posted to YouTube later tonight as well. I'm going to try and figure out what the deal is with that and why they're not showing up. Okay, so with the urinary system, okay, it's these four, or like I guess, I think of it as four because we've got our two kidneys, the ureters, and then the bladder. But really, it's kind of three discrete structures, right? So we have the kidney, the ureter, and then the bladder. I kind of tend to think of the urethra as just an extension of the bladder, even though that's not necessarily anatomically correct because they're made of different kinds of epithelial tissue. But that's kind of how I conceptualize it. But the urinary bladder is going to be the main storage component of urine. So believe it or not, some of you may not be aware of this, but the kidneys don't actually store urine. They're not supposed to. And usually that's really bad if they're storing urine. So the bladder is the main storage unit. And, as that fills up, we've got various kinds of responses that allow us to urinate or hold our urine if we're able to. And then the ureter here is the connecting vessel from the kidney to the bladder. So the ureter sends urine from the kidney to the bladder. Okay. Now, inevitably, this always happens every year. It probably won't be as big of a problem now because you won't have to write it out or anything. But usually in like a normal class setting, what ends up happening is people always confuse ureter and urethra. And you may just confuse them like if you're looking at a multiple choice question. But just remember that the urethra is the main vessel that exits the bladder, and the ureter is the contributing vessel from the kidney to the bladder. Okay, so ureter comes first, urethra comes second. It's the last vessel. Okay. Yeah, you're welcome, Catherine. And thank you to Kayla for mentioning that too. Okay, so where the heck are the kidneys, right? So the kidneys are in kind of an interesting spot in our body. So they sit just underneath the diaphragm and are kind of protected by our last two ribs. So ribs 11 and 12 are floating ribs, kind of hang out and, I'll put this in quotes, protect the kidneys from, some minor blunt force trauma. But they sit just underneath the diaphragm and kind of back behind most of the organs of the abdomen. So we can see here we've got all of our, our stomach, pancreas, the duodenum, intestines, all of that stuff has been removed. And now we can see that we have the kidneys that sit kind of right side by side with the major descending vessels of the abdomen. So we've got our abdominal aorta and the inferior vena cava that are passing right by them, okay? Now, it's also kind of interesting too, and this is more anecdotal, but the position of the veins relative to the arteries is actually flipped for how it is in the rest of the body. Most of the time, the arteries sit on top of the veins, like we can see down here with our, common iliac artery. Wow, hadn't thought about that in a while. Common iliac arteries, so our right and left common iliac arteries here sit on top of the right and left common iliac veins right here. So, but notice here that the renal veins versus the renal arteries, the veins sit on top of one another, or sit on top of the arteries rather. I mean, it's kind of anecdotal. Like I said, it's not necessarily like a test-worthy note, but it's something that's just kind of interesting is, some of this variation in human anatomy. So the kidneys sit just here, kind of right back behind the spleen as well. And we've got our little adrenal glands that sit up here, which again, the adrenal glands aren't part of this because they're part of the excretory system, even though they sit right on top of the kidneys. So we can see that the kidneys have their little connections with the ureters right here as the ureters lead down into the bladder. And what's kind of interesting too is, we'll circle back to this at the end of the lecture today, but the position of the ureters as they connect to the bladder is also interesting, and it's, kind of by design as well. As the bladder fills up, it can actually occlude or block off the ureters, and that's important for things later on. So when we talk about the position of the kidneys, right, the position is interesting because they're not technically in the same body cavity as the rest of the organs of the abdomen. Crystal, yeah, so the hilum is just the indentation of the kidney right here. So it's kind of a term that we use to describe just a little indentation, or kind of like, we use the word fossa to describe an indentation in a bone, or an articular facet or something like that, right? The hilum is just the little indentation where we have all the vasculature of, in this case, the kidney that is going to be entering and exiting the organ, right? So that's what the hilum is. So in terms of positioning here, this is interesting because, like I was saying, the kidneys don't exist in the same body cavity as the rest of the organs of the abdomen. They actually are what we call retroperitoneal, or more specifically, primarily retroperitoneal. So what does that mean? Well, when we say retroperitoneal, that means behind the peritoneum. And if you'll recall, the peritoneum is the lining of the body wall inside the abdominal cavity. So if I were to cut one of you guys open and open up your abdomen, you'd see this shiny film on the inside of the body wall if we, pulled your abdominal wall apart. And so that's the parietal peritoneum. The kidneys sit behind that. So they are what we call primarily retroperitoneal. So if we look here, we can see that this blue bit right here, all of this blue stuff is the parietal peritoneum. And we can see that the kidneys sit behind that. So here is the abdominal cavity, right? Our coelomic space. And then all of our organs, like our kidneys, as well as the vasculature of the abdominal aorta and the inferior vena cava, all of those structures are retroperitoneal. So they sit behind, or you can, if it helps you to think about it this way, underneath the parietal peritoneum here. Okay. So the cool thing about that is that these are not the only organs that end up being retroperitoneal in the abdominal cavity. However, what distinguishes these from other organs that become retroperitoneal later on is that these guys develop here and stay here throughout the embryological development period. Whereas some organs, like parts of the intestine, the transverse colon, for example, end up being repackaged and forced back into the abdominal cavity with so much force that they actually get pushed back behind and fuse into the parietal peritoneum of the abdominal wall, and then become what we call secondarily retroperitoneal. Okay, so this is just to highlight the position of the kidneys relative to the rest of the organs of the abdominal cavity. So they don't actually belong in the abdominal cavity. They're what we call retroperitoneal. All right. And what's kind of interesting too in this, when we would be doing dissections normally, what ends up happening is, we'll have students that cut into the abdominal cavity, and they, look and say, oh my gosh, are these tumors that the, animal or that the person, if we're working on a cadaver, has? And it's usually, sometimes there are tumors that develop. You find that a lot in animal specimens actually, but most of the time it's the kidneys, right? Okay. So now here we've got our longitudinal section through our kidney. Okay, so let's look at some of the features of the kidney. So the kidney has two major regions. It's got a medullary space and then a cortex. Okay. Now the division between those is going to be important because it separates and distinguishes different areas for solute concentration for things like water reabsorption when we talk about the functional unit of the kidney, which is called the nephron. So we'll get to that in a little bit. So we've got these two major areas, this inner area called the medulla, this medular cavity, and then this outer layer of tissue called the cortex, the renal cortex, right? And then the organ itself, the kidney itself, has this renal capsule that's not part of the cortex. It sits above the cortex, right? So what do we have inside the cortex? Well, we've got lots of vasculature that permeates the cortex here, but we also have about half of an organ that we call, or not an organ, but half of the functional unit of the kidney, which is what we call the nephron. And the nephron does all of the filtration of our bodies. So any sorts of salts or nitrogenous waste or anything that we need to excrete and get out of our body. It can sometimes not even be things like nitrogenous waste or salts. It could just be water, depending on how hydrated we are, right? So the cortex houses, some of the functional units of the nephron. It also has a rich network of arteries and veins that we'll look at here in a little bit. Inside the medullary cavity, however, we have some of our other structures, things like our renal pyramids. So the renal pyramids are formations of these vessels that are part of the nephron that we call collecting ducts. And these are what carry all of the extra filtrate, and at this point it's what we would call urine, carries the urine down into this little space that we call the calyx, okay? So the calyx is a little open sinusoidal cavity that basically holds the urine for short periods of time until it all collects and then is forced out of, the hilus, or this indentation. So this is what you were asking about earlier, Crystal, but the hilum here is just this indentation where you have all these vessels entering and exiting the kidney. But the renal pelvis here is where we have all of these calyces, of which we have a major and a minor calyx, or calyx, that all come together and form the ureter. There's smooth muscle that makes up some of the epithelial tissue that surrounds the ureter, and we have peristaltic contractions that can move urine from the kidneys down to the bladder there. Okay, so we've got our renal pyramids, we have our calyces. These renal papillae as well are these little areas where we have the openings of the collecting ducts as well. So, and then we have these little sinusoidal spaces as well, which kind of are just these areas where the calyces come together and form that sinus. All right. And then we have these columns in between each of the renal pyramids. And these columns are important because they bring with them or take away all of the blood supply to the kidney, right? Okay, so here's what it looks like in cartoon form, and here's the real thing, all right? So we can see we've got our renal pyramids right here, and we've got our columns that are spaced in between them where we have all of our vasculature taking place. So the renal pyramid, the calyx here, so the renal papilla kind of exiting there, and then we have the renal sinus. All of that comes together through the renal pelvis and then out through the ureter, okay? Then we have the renal vein and the renal artery. Renal vein, again, bringing blood away from the kidney. Renal artery bringing blood into the artery. And the distinction between the medulla and the cortex here. It's not as apparent, but you can use just the tops of the renal pyramids here as kind of your border of where the medulla stops and the cortex begins, or vice versa. Okay. All right. So here's where we get to the kind of one of the coolest parts about learning about the kidneys and the filtration systems of our body is the nephron, right? So the nephron is this really cool little set of tubules and specialized arteries that really their main goal is to first and foremost respond to pressure changes in our body. So things like if our blood pressure goes up or down, we have chemical and hormonal signals that will trigger different kinds of responses to make us either drink water or, feel like we don't need to drink water, or produce more urine, or filter more waste out of our bloodstream, right? All of these things kind of take place in the nephron. So the nephron itself is kind of divided up into a couple different components. So the nephron has this little starting that we're going to kind of start and imagine if we were a particle or some red blood cell passing through the nephron, or actually let's not think about it as a blood cell. Let's think about it, if we're a piece of, like, if we're like some sort of electrolytic salt or something like that needs to be filtered out of our body. So if we're in our bloodstream, we're going to be passing through some of our renal arterioles here, and we're going to enter the first component of the nephron, which is the renal corpuscle. The renal corpuscle is a composition of two major things. The first thing being the distinction of the arteries themselves. So the renal arteries, they kind of transform into these arterioles that we have what's called an afferent branch, and then the afferent branch is what's entering the renal corpuscle, and then the efferent branch is what's exiting. So A for away, E for exiting, okay? So this little bed of capillaries then gets a specialized name that we call the glomerulus. So the glomerulus is where all of our filtration is going to be occurring. So things entering and exiting the bloodstream into the kidneys starts here, right? And then we have this porous structure that surrounds the glomerulus and actually kind of has a basement membrane attachment to it through the basal lamina that we call the Bowman's capsule. Now, the Bowman's capsule is just a specialized branch of the rest of this tubular system that we'll get to in a second here. So it's not necessarily like completely separate or anything like that. But this renal corpuscle here is the combination of the glomerulus and the Bowman's capsule. So all of our filtering of our blood, of our immediate bloodstream, happens here within the renal corpuscle. Once the filtrate has exited the bloodstream, it enters our first division of the nephron called the proximal convoluted tubule. So it's a tubule because it's a tube. It's convoluted because it's always got all these like little twists and turns and stuff. And then it's proximal because it's closest to the start of the nephron, all right? So as the filtrate passes through the nephron, we'll talk about this more, but around 180 liters of filtrate pass through all of our nephrons every single day, right? And then at the end, once all of that stuff has passed through, then we're left with only about 1% of that initial 180 milliliters. So 180 liters. So about 1.8 liters is left over, right? And so that's going to be what we excrete as urine. So after our first stop or our first journey through the proximal convoluted tubule here, then we get down to this bit called the loop of Henle. And we'll talk about the loop of Henle in more detail later as well. Loop of Henle is pretty cool. This is going to be where a lot of our water reabsorption occurs. We end up reabsorbing about 65% of the filtrate in the proximal convoluted tubule into the cortex of the kidney. And, we'll talk about kind of where that goes in a little bit. But that filtrate that gets reabsorbed is important for controlling osmotic balance. And so understanding and kind of remembering what osmosis and osmotic balance is important, because that's what's going to determine where a lot of the water flows in our body. Wherever our salt concentration is, that's where water wants to be. So what we can do is we can have these, and not only that, but also making sure that the vessel our filtrate is passing through is permeable to water. The loop of Henle is super permeable to water. So we have this descending limb right here where most of that reabsorption of water takes place. So if we're super dehydrated, then the descending limb is reabsorbing as much of that water as it can into the medular space. Or if we're fairly well hydrated, then that water can kind of stay put. And so how it stays put is really dependent on what the salt concentration is of our medulla. If it's fairly dilute, then the water is going to want to stay in the loop of Henle. If it's pretty concentrated, meaning that there's less water there, then water wants to go there. Tying it back into what I talked about at the beginning of the year when we talked about, concentration gradients, that's all this is, right? So then the ascending limb here is kind of interesting too, because it's going to be important for determining where certain kinds of salts are being transported in or out of the ascending limb to kind of, again, modulate the concentration of the medular space right there. And then we have our distal convoluted tubule. So this is the last little bit of filtration where we can have, depending on how dehydrated we are, we can have up to 19% more filtrate that gets reabsorbed into the cortex there. So as the filtrate ends up getting back up there, it's usually fairly dilute or lower in concentration. And so again, depending on what that looks like, then the presence of something like vasopressin or antidiuretic hormone can control whether or not we need to reabsorb more water or get rid of some, right? So then finally, if we're a filtrate here, we've now passed through the gauntlet of the nephron. We've gotten through the renal corpuscle here, proximal convoluted tubule, loop of Henle, and then our distal convoluted tubule. And then finally, we end up in the collecting ducts where we can have multiple nephrons contributing to the same collecting duct. So all these little branches here are the would-be connections to the distal convoluted tubules of other nephrons. Until then finally, we end up all the way down here, basically just emptying out of this giant trash chute into the major calyx of the renal sinus there. So that's kind of our general journey of a piece of filtrate through the nephron, okay? So now let's look a little bit more closely at the renal corpuscle and kind of see what some of this stuff looks like, all right? So if we zoom in here, we have our little glomerulus that has some cellular connections with the Bowman's capsule here. So we'll look at some of these in a little bit more detail in just a second, right? Okay, so here we have our glomerulus, we have our afferent arteriole where blood is flowing into our nephron here, and then we have the efferent arteriole where blood is flowing away. So all of the blood that is passing through the efferent arteriole here has already been filtered, but everything that's passing through the afferent one still needs to be filtered here. These little cells that you can kind of see peeking out right here that sit around the afferent arteriole are what we call juxtaglomerular cells. It's like one of my favorite things to say is the juxtaglomerular cells. The juxtaglomerular cells are important because they are what respond to changes in blood pressure, and they can trigger a hormonal response to the hypothalamus to tell the hypothalamus to produce more things like antidiuretic hormone. Okay, so that's what this part looks like. And then we can zoom in a little bit more on these afferent arterioles and what this glomerulus actually looks like close up. So here we can see these juxtaglomerular cells present right here. So the juxtaglomerular apparatus is not only the juxtaglomerular cells, but also this macula densa that's present on the distal convoluted tubule. So like I said, these cells have specialized neural receptors that respond to changes in blood pressure. So what things can modify blood pressure? Well, blood pressure is just how much force is being pushed out on the internal side of the vessel here. One of the main ways to do that is just put more fluid in there. And if we have more fluid volume, that's usually an indicator that we're more hydrated. So if they are, pretty well stimulated by having blood pressure, being fairly high, then they can send a hormonal signal to the brain, to the hypothalamus, to have the hypothalamus produce antidiuretic hormone so that we stop producing urine, because it means that we're already super hydrated. So we don't need to be, we would need to produce more urine because we're already very hydrated. So the urine that we produce would be very dilute as a result of that. But if blood pressure drops, then we actually end up stopping the production of the antidiuretic hormone, because then we need to make sure that we're retaining as much water. So therefore, the urine that we produce is going to be much more concentrated as a result of that. All right. So let's look at some of the things that are going on here with our capillary bed inside of our Bowman's capsule. So what you'll see here that surrounds these little arterioles are we have all of these little things called podocytes. So podocytes are interesting because they're these dedicated cells that are pretty much kind of, you can associate them with like the visceral layer of the Bowman's capsule. So these guys kind of graft around the arterioles of the glomerulus. Now these arterioles are actually a little bit different than most other arterioles because these ones are fenestrated, meaning that they have little holes in them, right? So these little tiny holes are how we get filtrate out of our bloodstream and into the Bowman's capsule. But these podocytes are interesting because their goal is to create these little slits that can kind of monitor and control how much filtrate gets out and what the size of those filtrates are that get out, right? So this helps make sure that we retain certain kinds of blood protein, but that we can also get rid of other kinds of proteins that can fit through these little filtration slits. Okay. So what's interesting about these podocytes with those slit formations is that there's a correlation between congenital kidney failure and the malformation of those fenestration, or not the fenestrations, but of the filtration slits that the podocytes are responsible for. So typically what ends up happening is there's some sort of a genetic defect in the protein that forms the structure around those slits. It's usually one of two proteins called nephrin or CD2AP. So any sort of defects in those can usually cause some sort of acute kidney failure or a delayed response to kidney failure later in life. So that can happen if the podocytes aren't functioning the way that they're supposed to. Your book doesn't really go into a ton of detail about what podocytes are, so did a little bit of digging today and kind of figured some more of that stuff out. So that's the main function of podocytes, is that they help keep those filtration slits open and can kind of control the bulk size of molecules that exits our bloodstream. But then we also have these little filtration openings called fenestrae right here, so we can have, we can force different kinds of filtrate through those openings as well. Okay, so before we get to kind of how we control filtrate, let's look at some of the blood flow of the kidneys here. So if we start at number 1, if we're a blood vessel going into the kidney, we start out as the renal artery right here. So the renal artery then differentiates into an artery that we call the interlobular artery, which makes sense because the first branch right here goes into that interlobular space. So we call it the interlobular artery. Then once we get past that interlobular space, or the renal pyramid, then we get into the medullary area right here, where we specialize and become something called an arcuate artery. And then we have something called interlobar versus interlobular. That is a distinction there. So interlobar, between the lobes right here that we see in between these renal pyramids here. And then the lobular branch branches out into the medulla, going up into the cortex right here. right? So this interlobular artery has branches that go and differentiate into the glomerulus. Okay, so we've got our afferent and efferent arterioles, all right? So then this efferent arteriole, its job isn't over yet, all right? So we still need to vastly perfuse the area of the loop of Henle and each of the proximal convoluted tubules with these paratubular capillaries right here. That's number 8. So these paratubular capillaries are important because they can control some of the filtrate and where that goes if we're able to reabsorb it, depending on how hydrated we are. And then arguably most importantly, down here in the loop of Henle, where we have a lot of our water reclamation occurring, we have our vasa recta capillary bed. So this is a specialized capillary bed that's crucial for retaining water, or if we are really dehydrated, we can reclaim that water there and get it immediately back into the venous return system, okay? So the vasa recta then differentiates into or contributes back into the interlobular vein, okay? So interlobular vein, arcuate vein, interlobar vein, and then back out through the renal vein, out through the renal hilus, okay? So that is our blood flow trajectory going from the renal artery all the way through and then back out the renal vein. Okay, so what controls whether or not we can move filtrate from our bloodstream and get it out of our bloodstream, right? And a lot of it's just pressure, okay? So it's the same idea where we have, concentration gradients kind of ruling everything. Here, we just have pressure gradients, right? So we've got 3 major kinds of pressure that control this. We have the glomerular capillary pressure, which is generally the highest one, all things kind of being normal. This is the pressure that the afferent arteriole exerts into the wall of the capillary here as we get down into the glomerulus. And this is generally much higher than what we have called the capsular pressure and then the colloid osmotic pressure. So the capsular pressure is just the pressure inside of the Bowman's capsule right here, just by virtue of its existence. And then we have the colloid osmotic pressure, which is interesting because this can typically vary depending on different kinds of macromolecule content inside the Bowman's capsule. So what do I mean by that? Well, things like protein content can affect things like the colloid osmotic pressure. So for example, if you have a diet that's really rich in protein content, then what you might notice is that urine production typically goes down because the colloid osmotic pressure in your nephrons goes up because all of the protein that's present in your bloodstream ends up getting secreted and becomes a significant part of the concentration of the pressure inside the Bowman's capsule. So that colloid osmotic pressure increases with things like increased protein consumption. So that ends up resulting in a greater colloid osmotic pressure that, depending on the situation, can sometimes outmatch or get really close to the glomerular capillary pressure. It won't ever, it could theoretically outmatch it if you overconsume protein and you're not producing any urine at all. And so then what'll end up happening is all of the filtrate kind of ends up going back into the bloodstream, and then that's bad news bears. But so colloid osmotic pressure can vary and get closer to the pressure of the glomerular capillary pressure, right? And so depending on these pressure gradients, that determines how much urine we end up producing, okay? So the lower these things like our capsular pressure and colloid osmotic pressure, then the greater our urine volume is because the glomerular capillary pressure is forcing a lot of that filtrate out and contributing to a lot of that volume that's passing through the nephron. Okay, so how do we make urine, all right? Well, we've got 3 major steps that are part of the urine formation process. So the first part is the filtration bit, which we just talked about. And then we have the second phase, which is the tubular reabsorption phase, or sometimes people say resorption, but reabsorption, where we can either reabsorb water or any kind of extra salts that we need from our excretory system. So sodium, chlorine, potassium, hydrogen even as well. We can reabsorb and get any sort of extra ones that we need from the filtration system and get them back into the afferent arteriole. I'm sorry, not the afferent, the efferent arteriole here. So this is great because it's kind of a way that we can kind of check ourselves if we don't have, or if our body has excreted too much of one thing, then since these arteries pass by the filtration system, they can kind of, check and make sure that there's not anything else that we need. It's like, are you sure we got rid of that? Do we need it? Are we good? Do we not need it? Then cool, then we can get rid of that stuff if we don't, right? And then finally, we have the last bit, which is our tubular secretion. So anything that we absolutely don't need, and again, generally that's due to greater osmotic potential. All of our solutes get secreted via the green arrow. They get put into the final collecting duct, which is where kind of our urine is synthesized. This is our most concentrated form of our nitrogenous waste. And even at our most dehydrated, or in general, the urine content that we produce is only about 10% urea, and the rest of it is water. All right. So I've already kind of talked about this kind of by proxy, but we'll still go through it anyway. But this is the main mechanism that controls how we concentrate and how we basically produce urine. And all it has to do is with concentration gradients, right? So on the left-hand side with this figure, what we're seeing here is the relative concentration of the interstitial fluid measured in milliosmoles. All right, so what does that mean? Well, for those of you who don't know, basically milliosmoles are just standardized measuring units, kind of like milliliters and liters. These are just measurements of concentration. Usually when we measure concentrations of things, it's measured in moles, but these are millimoles. So think of it as like, volume is in liters versus milliliters, concentrations usually in moles. And then in this case, since it's quite small, we would call it millimoles. So the milliosmotic potential is just an indicator of what kind of net movement we would see of solutes, or in some cases water, out of the tubular system. Okay, so in general, if a number is bigger, okay, then we would end up seeing the net movement of something out of the tubular system. And generally, if the number is smaller, then we see movement into the tubular system. Okay, so when we start out, we're fairly low concentration here in the cortex, right? But as we move from the cortex to the outer and then subsequently the inner medulla, the concentration of this interstitial space goes up. So when we talk about concentration, what are we talking about? We're talking about the concentration of salts and other kinds of electrolytes. So potassium, sodium, chlorine, hydrogen, all of these things are going to be in greater concentration as we move down through the tubular system, okay? So about 65% of that water gets reclaimed up here in our proximal convoluted tubule. And then once we reach our descending loop of Henle right here, this is important because we can retain a lot more water if we need to, right? So upwards of 15% more of that filtrate content can be reabsorbed purely as water, not even factoring in things like sodium or chlorine or anything like that. So how do we control for those kinds of salts? Well, the epithelia that makes up this descending loop right here isn't permeable to anything but water, which is kind of nice. It's like having a specialized filter just for water that water can enter and exit through. And what's really great too is not only are these epithelia not permeable to things like salts, but also depending on the concentration, especially of the inner medulla, on how hydrated we are, then that's also going to determine where water moves. Since there's a lot less water out here in the interstitial space compared to the inside of the tubular system here, then water wants to be where there's less water. Water is just moving down its own concentration gradient, okay? Then as we move through the descending loop up the ascending limb here, then we can see there, depending on what we need to do, we can excrete more salts. So if we need to reclaim more water, we can put more salt out into the interstitial space here where we can reabsorb more water. Or if we're already fairly hydrated, then we can make sure that we don't need to, secrete any more sodium or chlorine or anything like that. And we can continue to do that up through the ascending limb up here. And we can see that we go from the thin to the thicker portion right here. All right, and so the epithelia will change as we get farther up the ascending limb. So then as we reach the distal convoluted tubule up here, so this is where our urine is getting to its most concentrated point, right? So the distal convoluted tubule and collecting ducts are permeable to water only if antidiuretic hormone is present, right? So if antidiuretic hormone is present, that means, hey, we're super dehydrated. We need to be, reclaiming as much water as possible. So then that allows for water to escape the distal convoluted tubule so that we can reclaim that back in the cortex up here, okay? So water can exit, and then as our filtrate exits out through the collecting duct right here, we can lose and retain more water if necessary, generally whittling it down to about 1% of the total volume that passes through there, so about 1.8 liters of the 180 liters that we see passing through there every day. All right, so that's how we concentrate our urine. Just as a fun little side note, so at the time of recording this, the new Dune movie is coming out pretty soon. I don't know if any of you guys are into science fiction or anything like that, or if any of you guys have read the books, the Dune books by Frank Herbert. Thumbs up if you have. Anybody read them or know what I'm talking about? No? Nobody? Oh, did you see the first one, Monteith? I haven't seen the first one. I've heard it's pretty bad. Yeah. Oh, nice, Daniel. Heck yeah, dude. Oh, not Doom. Dune. D-U-N. Here, I'll put it in the chat. Yeah. Sorry. So yeah, Dune. So I bring up Dune because not only is so the books are, so Daniel, you might hate me for saying this since you're reading for the third time. I'm not a big fan of Frank Herbert's work, and we can debate that at another time if you are at odds with me on that. But the books themselves, to put it as a long story short, it's basically about this family that ends up moving to a desert planet where water reclamation and water and just water in general are sacred. And one of the main characters, named Paul, ends up kind of going on this quest to kind of I won't spoil it or anything like that. You guys should watch the movie. It looks pretty good. It's supposed to be, I think it's supposed to be pretty good. But I bring it up because there's an animal that's mentioned in the books called a kangaroo rat. And I won't spoil it or anything, but kangaroo rats play an important role in that book series. So why are we talking about kangaroo rats? Well, kangaroo rats live in the desert. We have them. They're kind of local to our area. They're endemic to our area. But they're also a protected species as well, especially the desert miriami species that's local here. But what's cool about these little furry T-Rexes, amongst a host of other kind of really cool muscle physiology things, they have these hyper-developed loops of Henle. So the cool thing about kangaroo rats is they don't actually drink water in their nature. It's not in their nature to do that. What they do get is they get a lot of their water from different kinds of fruits and nuts from what they forage for. And since their loops of Henle are so well developed and so highly vascularized, they're able to reclaim almost 100% of the water that they consume with their food. So they're nearly 100% self-sufficient with water reclamation in their own kidneys. It's really, really cool. I don't know if this is, scientifically supported, but what I would imagine is people who grew up in deserts or are endemic to arid climates and are used to surviving on lower amounts of water very likely have physiological adaptations for living in areas like that. One of them could be things like more developed loops of Henle within their kidneys. Kind of neat to think about. But it is well studied and well fleshed out in kangaroo rats. And if you don't know what a kangaroo rat looks like, Google it. And then read the Dune books, because then you get to find out what role kangaroo rats play in that series. Kind of neat. Okay. So here in the next couple slides, we're going to talk about or look at kind of what reabsorption in each of these parts of our tubular system looks like. So here we've got reabsorption in the proximal convoluted tubule. So here we've got a couple of different colored lines. Red means active transport, green means co-transport, and then orange means osmosis. So the red and green arrows are going to indicate where salts are going to be getting reabsorbed back into either the bloodstream or into the interstitial space. And then orange is just going to be the movement of water. So red and green, things like active and co-transport. Active transport would be for things like different kinds of salts, like sodium, chlorine, potassium, those kinds of things. Co-transport is usually reserved for things like different kinds of buffers, like bicarbonate, but also things like sugar molecules, like glucose and fructose. Okay, so all of those things can be transported through these cuboidal epithelia of the proximal convoluted tubule into either the interstitial space or that peritubular capillary, which is just an extension off of the efferent arteriole. Okay, so now we get down into the loop of Henle. So we've got our descending limb and kind of the thin segment of our ascending limb here, right? So what we've got going on here is we have our simple squamous epithelia. So it's going to be a different epithelial content within the descending limb versus the ascending limb here, okay? So again, this thin, simple squamous epithelia, nice and permeable to water. And again, depending on the salt content within the interstitial space, that can determine the magnitude of the water reabsorption that we see here. As we go up the ascending limb here, what we end up seeing is that water permeability decreases, it goes down, okay? So the thin segment of the ascending limb of the loop of Henle is not permeable to water. However, it is permeable to solutes. The solutes diffuse out of the tubule and into the more dilute interstitial fluid as the ascending limb projects towards the cortex. The solutes diffuse into the descending vasa recta. So if we need extra salt content in the bloodstream to modulate things like blood pressure, then we can have that occur within the ascending limb of the loop of Henle. Okay, so then back to our red and green lines here. So with the ascending limb, we see only salt permeability, no water permeability present here for these cuboidal epithelia. So here we're excreting things like sodium, potassium, chlorine, those kinds of things can be excreted into the interstitial space or into the vasa recta. Okay, and then finally, through the distal convoluted tubule as well as the collecting ducts here, we can, if necessary, resorb more water, with the presence of ADH. And then we can also reabsorb more sodium, more chlorine, more potassium, anything else that we need. So what's kind of funny is you might be thinking, well, if we're trying to get rid of all of this stuff, then why the heck are we bothering, reclaiming it in the first place? And recall that the filtration that occurs at the glomerulus level is not specific to things other than size and pressure. However much pressure we have there is going to determine whether things get excreted or pushed into the filtrate or not. If pressure is down, then not a lot of filtrate forms. If pressure is up, then a lot of filtrate forms. So think of the tubular system as kind of like an extra way to make sure, okay, we're not getting rid of any extra things that we don't need. So it's also a way that we can catch that, but it's a way that we can control whether or not we need to reclaim more water as well. Okay, so now here we're going to look at a couple of different hormonal pathways as well, so different kinds of hormones that the kidneys can produce and their effects on the rest of the body. One of the simple ones is the kidneys can produce renin. So renin gets produced in the kidneys, and that gets produced or released into the bloodstream, which then triggers a angiotensinogen to angiotensin pathway utilizing the lungs. Angiotensin 2, if you recall, ends up kind of coming back around and increases the production of aldosterone within the cortex of the kidney. The reason that's important is aldosterone can then act on the nephrons of the kidney to allow for increased sodium and water reabsorption and help increase things like blood pressure. So one thing that you'll find here is hydration, dehydration, all of that stuff is generally monitored or regulated in our body by things like pressure, also pH, but a lot of it's pressure. So if things like concentration of blood, or if blood pressure drops or something like that, or if blood pressure raises, then we have all these hormonal pathways that kick into effect to help make sure that we, have a normal blood pressure, and that we can, get rid of water or get rid of salts if we need to, or reclaim them if we need to. So if we need to reclaim a lot of salt, then we can do so, and then simultaneously we can reclaim more water to help increase blood pressure, okay? So antidiuretic hormone, okay? So sometimes, like I said, what can happen is we can have increased blood solute concentration or a large drop in blood pressure. So that triggers different kinds of neuroreceptors, specifically baroreceptors in places like the aortic arch, the carotid sinuses, different places in our body will have these specialized receptors. That sends a signal to the posterior pituitary gland that's dangling down from the hypothalamus. It goes to the hypothalamus first, but the hypothalamus then has neurons that trigger the release of that antidiuretic hormone. Antidiuretic hormone increases water reabsorption and results in decreased solute concentration and increased blood pressure. So we offset the initial stimuli here. So we're you can probably tell we're getting around to looking at another homeostatic mechanism here. And then finally, we have another kind of hormone that gets produced by the heart that has similar results to things like to ADH. Atrial natriuretic hormone can control the permeability of sodium and water with response to blood pressure. So typically what happens is the initial stimulus is an increased blood pressure in the right atrium, okay? So the right atrium can then trigger a response for atrial natriuretic hormone, which then acts on the kidney. What this does is increases sodium excretion and increased water loss, resulting in a drop in blood pressure. So if we are overall too hydrated or blood pressure goes up, then we can excrete water from our body or excrete extra salts this way. So just as a kind of a quick aside, there's certain chemicals that can make us form urine at faster rates, and we call these diuretics. We have different kinds of diuretics that can have different kinds of clinical implications. Some of the more common ones, especially for Western society, are these ones that kind of imitate atrial natriuretic hormone, which help increase the permeability of salts into our tubular system so that we can get rid of extra salts and we can flush out our body if we need to. So typically these ones, I forget what they're called. If you guys know it, you can remind me or write it in the chat, but there's these special diuretics. That's a bummer, it's escaped me right now. But these specialized kinds of diuretics that can help flush sodium out of our body if we need to. So things like hypertension, kind of precursors to heart attacks, those kinds of things, are sometimes predicated on elevated levels of salt and increased levels of blood pressure. So this is why, kind of tying it back into, monitoring different values of blood pressure, one of the more important values to look at is the systolic pressure. So the systole pressure, the active pressure of the ventricle against the arterial wall. So the reason that's important is because the ventricle and the health of the ventricle is going to determine how much force it's putting on the walls of the artery. And so if that value is getting higher and higher, then that's, usually not good. And so, the way that you can kind of control that and bring that down is with these different kinds of diuretics that can help control that, along with some other chemicals like beta blockers and stuff, but they're basically mimicking this natural hormone that we produce. That's why I brought that up. Okay, so if we look at different kinds of homeostatic mechanisms, so this one is looking at blood volume, okay? So if blood volume increases or decreases, right? Typically, when we think about blood volume, especially in our vessels, another easy way to think about that is with respect to pressure. Because if you have more volume, then you're going to be exerting more pressure on the arterial walls, okay? So elevated blood pressure, baroreceptors in the posterior pituitary can inhibit, baroreceptors inhibit posterior pituitary ADH secretion when blood volume increases. So we need to be producing urine, getting rid of this stuff. The juxtaglomerular apparati inhibit renin releases when blood volume increases, which decrease aldosterone secretion, again, making sure that we produce urine. Atrial cardiac muscle cells secrete atrial natriuretic hormone when blood volume increases, so we can get rid of that extra volume. And then the blood vessels themselves, sympathetic division baroreceptors detect increased blood volume, which causes vasodilation of renal arteries, right? So we can increase the size of those blood vessels to help compensate for that pressure. Because if the pressure is too high, then we aren't going to be able to, get any of this filtrate out through the tubular system. And then we can have the opposite effect happen if blood pressure drops, or another way that we can have a similar response occur is if we have an increase or an elevation in the concentration of solutes in the bloodstream. Okay, so in the interest of time here, we're going to kind of move right along. So once we've gotten to the urine part, how the heck do we get out of the kidneys, right? We've only talked about the kidneys so far. So now let's get into the ureters and the bladder, right? So the ureters, okay, have a collection of smooth muscle cells that surround them, but we have these specialized kinds of epithelia that we find only in the ureter as well as in portions of the bladder that are called transitional epithelia. So these transitional epithelia are important because they can expand fairly easily, but also contract and resume a fairly constricted shape if they're not, full, or they don't have a large volume present in them. Okay, so we move from the ureters down into the bladder here. The bladder has a specialized portion that doesn't have transitional epithelia, but the rest of the bladder is made up of transitional epithelium. This trigon of the bladder here is composed of where we have two ureteral openings, and then we have the main urethral exit right here, okay, where we have an external sphincter that can help close off the opening of the urethra, and then when we urinate, that external urethral sphincter will relax and then allow urine to pass through. So this trigon of the bladder doesn't have transitional epithelia because what's important is that as the kidneys fill up with urine, we need to be able to have the bladder expand and have it block off the ureters. So you'll notice that as the ureters come down, they come down on the backside of the bladder right here. And again, like I mentioned, that's by design. So as the bladder fills up, then we, shut off, or we close off and kind of pinch the ureters closed so that we can't add any more urine to the bladder. And this usually only happens if your bladder is really, really full, okay? So that's usually an indicator that we need to go to the bathroom, okay? So as the bladder fills up and stretches, we have a signal that gets sent to the brain, okay? So ascending pathways carry the increased frequency of action potentials of the spinal cord to the pons and cerebrum. When the urinary bladder becomes stretched, this increases the conscious urge to urinate. So the reflex that gets sent back via somatic motor nerves right here, so we synapse here, is what's called the micturition reflex, okay? So that allows for us to unconsciously relax our external urethral sphincter, and then we can, urinate as normal. So the urine in the urinary bladder stretches the urinary wall. Action potential is produced by stretch receptors carried along pelvic nerves, the sacral region of the spinal cord, and then they are carried by parasympathetic nerves in the red line, okay, to contract the smooth muscles of the urinary bladder. Decreased action potential is carried by the somatic motor nerves in the purple line, cause the external urethral sphincter to relax. So this would be an example of a polysynaptic reflex where we're activating the parasympathetic nerves of the smooth muscles of the bladder, but inhibiting the somatic nerves of the external sphincter so that can relax and then contract simultaneously. So kind of akin to if you put your hand on a hot stove, the reason you can pull it away is because you're activating flexors and inhibiting extensors. It's the same idea. So just kind of a quick note on bladder health here. Sometimes what can happen is bladder tissue can become infected with cancer cells and can develop cancerous cells. Apparently it's a leading cause of cancer in the United States. But interestingly enough, one of the treatments for bladder cancer is actually an injection of live tuberculosis causing cells that ends up fighting off the cancerous malignant cells inside the bladder. And if done early enough, it can actually increase the rate of survival up to 70% over a 5-year period. So that's pretty cool. It's actually sort of similar in a way that we can use different kinds of bacteria to help outcompete and treat things like, different kinds of skin, acne conditions or things like that. And so I guess just as I'm thinking about it, one really quick thing with the ureter here, one of the reasons why it's important to void the bladder if you have a very full bladder is the urine can actually back up into the kidney and cause kidney failure. Kidney failure can manifest in a couple different ways through things like acute nephritis or chronic nephritis, where you have inflammation of the nephron. You can have complete degradation of the glomerulonephritis, which can end up resulting in foamy, blood being produced with the urine as well. Not good things. Lots of animals are actually prone to developing kidney disorders. Unfortunately, cats are. So, I love cats and dogs, but my wife and I have had cats for the past couple of years. And our first cat that we got suffered really severely from kidney issues. And the vets told us that, it was nothing more than we just got an unlucky roll of the genetic dice. So unfortunately, we had to put him down after a year because his kidneys would keep developing these stones because he kept getting blocked and his ureters would back up. And so it was a sad thing, but he's in a much better place now. And so, we got to have him for a year and he was awesome. But yeah, kidney health, it's a real thing. Okay. So what makes us thirsty? This is kind of just a culmination of all of the rest of the material that we've talked about today. But all of these receptors are present within the hypothalamus. So things like an increased level of blood concentration, so increased concentrations of salts or a change in blood pH can make us feel thirsty. And so one of the ways that our body knows that we can dilute that is if we introduce, a dilute solvent like water into our bloodstream. That helps bring down the concentration of whatever solutes we have in our blood, as well as fights the drop in blood pressure that we're likely experiencing at the same time. So again, through chemoreceptors and baroreceptors, we're able to send out a signal and tell us to drink water to help compensate for these things. We also not only have this thirst response or this thirst reflex that we develop, but we also have other kinds of chemicals like buffers that exist in our bloodstream. So we talked about those quite a bit with the respiratory system as well, with the importance of bicarbonate and a carbonate ion that help control the drastic pH swings that can happen if concentrations in the bloodstream rise. Because you don't want that to happen, because if you have drastic pH concentrations that are allowed to continue to increase, you can develop things like alkalosis, or if pH drops below a certain threshold for a prolonged period of time, then you can develop acidosis too. All right, let's see. We've got our final two homeostatic regulations here. So we have potassium ions, again, so potassium controlled by receptors within the cortex of the medulla of the within the cortex of the adrenal gland. With increased aldosterone secretion, what this ends up doing is increasing the rate of potassium ion secretion from the effector cells within the tubule system, the distal convoluted tubules and collecting ducts of the kidney, and getting rid of potassium in the urine. And then if we drop below certain thresholds of potassium, then we stop producing aldosterone. Okay, so then as I mentioned, pH, things like increasing water consumption, but also the effectiveness of buffers that are present in the bloodstream can help control pH values. So what might end up happening is if your pH is dropping or anything like that, or if it's increasing, then we can control things like our breathing rate, because that angiotensinogen to angiotensin pathway can help with increasing the regulatory capacity of those buffers that are present in our bloodstream. Okay, so that's it for today, you guys. Let me stop this here.